So, Assalamu alaikum. This is the fourth class, that is lecture number 4 for us, and we are here to learn about the cell and signal studies. And now we are going to start this new lecture. और इस लेक्चर में हम बहुत सारी चीज़ों को समझने की कोशिश करेंगे और हम ये देखेंगे कि क्या चल रहा है इस टाइम, इस वक्त नीट की क्लासेस चल रही है. स्टार्ट दिस न्यू लेक्चर और इस लेक्चर में हम just a minute, I am trying to fix this and would like to know whether this is running smoothly because there is some error showing here. Okay, now it is okay. Now the video is clear. You can watch it now. And now we are going to go for a live class session. Go for a live class session. In the last video, we have tried to learn about the PUC vector and PBRC vector. So this is very important for us to learn about the PUC vector and PBRC vector, and it's very important to understand now other types of vectors. So welcome everybody again. I'm just live now, and if you are watching this from any state of the India, it is very important for your NEET, and mostly the questions are asked from it. So first of all, we try to understand what are the bacteriophages. So the definition of the bacteriophages: the bacteriophages are the viruses. Now, first of all, we have to understand what are the bacteriophages, and the bacteriophages are the viruses. So they are viruses. So important thing is that we have to understand that they are simply a viruses. They are not the vectors, but we can use them as the vectors. So what type of vectors are the bacteriophages? They are the they are the viruses that replicate within the bacteria. That means the DNA of these bacteriophages replicates within the bacteria. So bacteriophages are the viruses. They are simply phages and they replicate within the bacteria. So some phages which incorporate the DNA into the bacterial chromosome and remain there permanently. And phage vector can accept shortened fragment of the foreign DNA in their genome. So this phage vector, this is the bacteriophage. This is the bacteriophage. The bacteriophage is having the DNA inside it, and this phage vector is important for it. Now, the bacteriophage, which is having the DNA inside it, and this when it incorporates into the bacteria, either it remains permanently there or sometimes it replicates. And there are two ways. If it will remain permanently there, then it will go into the phase of lysogeny phase. If it will replicate itself and make multiple phages, then it will go into the lytic phase. The lysogeny and lytic phase depends upon the when there is enough food resources. If the bacteria is on the less food resources, that means the less amount of the food is available for them, then definitely it will go for the lysogenic phase, and it will not replicate because the energy is needed, ATP is needed for their application. And if there is no ATP, there is no energy, what is the purpose of going for their application? So it will integrate itself. There will be the integration of the bacteriophage DNA into the bacterial DNA. So that is called as the lysogenic phase. Then now there is another type of phase that is called as the lytic phase. What is this lytic phase? In case of the lytic phase, the bacteria is having the enough food resources. The bacteriophage will replicate its DNA, and then will make it as protein, that is a capsid protein coat inside the bacterial DNA, and then the bacteriophage will lyse, and this lysis will cause the uh, breakdown of this bacteria and the killing of this bacteria. That's why it is called as the lytic phase or lysis. Now, another topic is what is cosmid. So the cosmids are also the vectors, and they are possessing their characteristics of both plasmid and bacteriophage. If we will bring some characteristics from the bacteriophage, and some of the characteristics from the plasmid, combine them, and they are called as the cosmid. So what are those characteristics which are present in, sorry, which are present in the cosmid? And we can say in short that the cosmid is vector is a hybrid of plasmid and bacteriophage, possessing best features of both. What are the best features inside the plasmid, and those best features inside the bacteriophage? We will combine them, and the new type of vector will be formed, that is as the called as the cosmid vector. So I will tell you in the another very soon. I will tell you about what are the foreign DNAs that are inserted into this. First of all, we will try to understand the basic structure and formation of these vectors. So these bacteriophage, now this cosmid possesses the features of the plasmid and bacteriophage. Cosmid grows in the plasmid from form in the bacteria. Yeah, that means they grow in the plasmid form in the bacteria, and they can replicate like plasmid and can be packaged like the bacteriophage. So what are the best characteristics? The best characteristics is the replication is like the plasmid, the way plasmid is replicating. The cosmid DNA is also replicating like that, but the way bacteriophage makes this packaging of this this vector, it is like the bacteriophage. So they can accommodate inserts of about chimeric DNA up to 50 kilobase pair long. That means up to 50 kilo base pair, one base pair, and then 1,000 base pair. That means 1,000 into 50, 50,000 base pairs will be the long DNA insert, that is chimeric DNA, can be inserted into the cosmid. Cosmid contains an antibiotic resistance gene that is useful in identifying the recombinant DNA, and they can also possess an origin of replication like the bacteria that like the plasmid is having the origin of replication. They can also have the mm, uh, uh, that is origin of replication and facilitate their propagation in bacteria, and also possess a cos site that helps in the packaging. The cos site is present in the bacteriophage that helps in the packaging of the this DNA, and that is why it is called as a cosmid. Now, the cosmid can be constructed by adding the fragments of the lambda phage DNA, including cos site, and to the plasmid. So the foreign DNA is up to, we have written here the 40 kb, and some books have written up to the 50 kb, and can be inserted in the cosmid. And recombinant DNA so formed can be packaged as the phase and injected into the E. coli. Once they go inside the host cell, cosmid behaves like the plasmid and replicates. So the behavior of the plasmid cosmid is like, when you for the replication is like the plasmid, and the advantage with the cosmid is that they carry large fragment of foreign DNA compared to plasmid. So what is the DNA that is carried by the plasmid? That is only 10 kb, and this bacteriophage carries 5 to 25 kbs. And then there is cosmid, and that cosmid carries up to 35 to 40, 45 or 50 kbs. And then there is plasmid artificial chromosome, that is pack, and that carries that is the host is always E. coli inside for them, and that carries the foreign DNA of 100 to 300 kbs, and bacterial artificial chromosome that is also carries to 100 to 300 kbs. And there is yeast chromosome, the host for that is cerevisiae, Saccharomyces cerevisiae, and that carries 2000 kbs of this forge. So first of then now we will try to understand about the yeast artificial vectors and what are the yeast artificial vectors. So first of all, we will try to understand the yeast vectors that have been engineered to contain a centromere. That is, they are having the centromere, and like the human chromosomes, they are having their telomere and the origin of replication. So yeast artificial vector was introduced in 1987 by Olson, and yeast artificial vector is synthetic DNA that can accept large fragments of DNA up to 300 up to 2000 kbs kilobase pairs. Now it is thus possible to clone large DNA piece by YAG. That means if we are having a large DNA piece that is 2000 kb gene and we want to clone it so as to get the multiple copies of this gene, this gene we can use the yeast artificial chromosome. Now another type of chromosome is the human artificial chromosome, and this human artificial chromosome was developed in 1997 by William Willard. And human artificial chromosome is synthetically produced. It is synthetically produced. Yeast artificial chromosome is also synthetically produced. Vector DNA possessing the characteristics of human chromosome. It is having the characteristics of human chromosome. What are the characteristics of human chromosome? It is self-replicating microchromosome, and its size ranges from 110 to 1/5 of the human chromosome. That means its size ranges from up to 110 to 1/5 of the human chromosome, and it carries relatively long human genes. Those genes which are very long human genes, it can replicate them and keep plays and it plays important role in the gene therapy. This is the question that can be asked in the NEET examination. Which of the following chromosomes can be used for the gene therapy? And these questions you need to remember that hack, which is the human artificial chromosome, can be used for the gene therapy purposes. So this is very important for you, and it's very important to understand what is the purpose of this hack. Now going further, there is another bacterial artificial chromosome that we discussed earlier. Bacterial artificial chromosome also carries the fragment of 300 kbs, and this is based on the F plasmid, which is larger than the other plasmids used as a cloning vector, and it can accept a DNA insert of 300 kbs. Now these are the five types of the these plasmids which we have understood in this class, and it's very important to understand what is the purpose of these mm, uh, chromosome, these are plasmids. And if the fragment, we need a smaller fragment of a DNA that need to be cloned, we can use the normal plasmid vector, that is PUC and PBR. And if we need the higher uh, sizes of these vectors, higher foreign DNA, then we use the yeast artificial chromosome, and depending upon the size of the foreign DNA. And if we need further highest base pair DNA, that is long DNA, human DNA that can be cloned, and we use the human artificial chromosome or yeast artificial chromosome, depending upon the size of the DNA. So that need to be understand, and that need to be know what is the purpose of each of these. Now these are the human artificial chromosomes, where you can see that they are having the telomere and high molecular weight human DNA, selectable marker, ligate the transcript. We added the human artificial chromosome here, and then it will be called as the mini chromosome as well. This is how we make the human artificial chromosome. Now this is the size that I told you that I will tell you what will be the host cell for each vector system, and what is the insert capacity, how much insert they can carry. It depends upon the present days measures, that how much warload or how much animation they carry and put it on onto the other country. And it is in the same way that we are having these vectors. So the plasmid, the host cell is E. coli, and if the host cell is E. coli, then the insert capacity is 0.1 to 10 kbs. 0.1 to 10 kbs. If the bacteriophage is there, then the capacity is 10 to 20 kbs. If the cosmid is there, then is 35 to 50 kbs. And if there is any other type of phosmid that is having the F plasmid, that is insert capacity is same 35 to 45 kbs. And bacteriophage P1, that is called as the plasmid artificial chromosome, or bacteriophage P1, that is having the capacity of insert capacity of 10 to 100 kbs. Just a minute, I will get this battery changed for this pen which I'm using for writing here. And you keep watching this for a minute. It will take few seconds. Now the pen is ready. uh, Thank you for waiting. Now this human artificial chromosome, you know that is 100 to yeast artificial chromosome, that is the yeast is the host, and it is 1000 to 2000 kbs, and human artificial chromosome more than 2000 kbs. Now we will try to understand what are the methods of the gene transfer, and then after that we will study the expression vectors and choice of vectors. First of all, we will try to understand what is transformation and how transformation is going to help us, how these vectors can be used for the cloning. Now, transformation is the method of introducing foreign DNA into the bacterial cell. So, how foreign DNA can be introduced? For example, this is a vector and this is a foreign DNA. How this can be introduced into the bacterial cell? So, the uptake of plasmid DNA by E. coli is carried out in the ice cold calcium chloride, that is 0.05. That means we need the ice cold buffer that which carries calcium chloride. Why calcium chloride? I will tell you what is the use of calcium chloride in the for this purpose. The calcium chloride is causes the opening of the pores. So what is the purpose of calcium chloride? Calcium chloride makes this bacterial cell competent and allowing them to take up the foreign DNA. So the purpose of this is to make the bacterial cell competent. Bacterial cell competent. So this is the first step, that we use a lower temperature and make the bacterial cell competent. And this calcium ion is neutralize the negative charge on the both DNA and DNA backbone and the lipopolysaccharide of cell membrane. So what is the purpose of these calcium ions? These calcium ions, that means the purpose of these calcium ions is, first thing we need to understand what is the purpose of calcium. And the calcium, it is calcium 2 positive, and the calcium ions that is having ions, they neutralize they neutralize the negative charge. They neutralize the negative charge. So this is the first function of them, that they neutralize the negative charge. Where is this negative charge?